Morning students, today we are going to see about the next pathogen, that is Escherichia coli, nothing but E. coli. You already know about the basic characteristic features of E. coli, but let us discuss in clear. E. coli, that is Escherichia coli, is a gram-negative, motile, facultative anaerobic rod-shaped bacterium. It is one of the most common hospital acute pathogen, and it is main reason for the cause of urinary tract infections, abdominal infections, and bloodstream infections and all. And it is a gram-negative facultative anaerobic rod-shaped bacteria, and it is one of the commensal. Commensal means the species which gets benefit from the association with the other species, and benefits means it may get nutrients, shelter, support, or even locomotion from the host species. So E. coli is one of the commensal, and it mostly found in the lower intestine of warm-blooded animals. A small proportion of this E. coli are pathogenic. It means not all the genes or strains of this E. coli are pathogenic. Some strains of E. coli are pathogenic. The harmless strains which produces vitamin K, and it prevents the colonization of this bacteria, pathogenic bacteria in the intestine. And this is the structure of E. coli, and it is having the DNA and outer cytoplasm with a cell membrane and flagella. Mostly E. coli is having peritrichous flagella, and it is rod-shaped bacterium. And the length of this E. coli is up to 1 to 3 micrometer, and width is 0.4 to 0.7 micrometer. Maybe if we see in microscope means the E. coli are arranged single or in pairs, and it is having peritrichous flagella, and some of the strains are non-motile also, and it is not producing any type of spores, and capsules and fimbriae are also found in some strains. The classification of E. coli is based on the serotypes, which is having the cell wall O antigen and capsular K antigen, fimbrial F antigen, and flagellar antigens also. Cultural characteristics of E. coli: it is an aerobe and facultative anaerobe, so it requires oxygen, as well as it can grow in the absence of oxygen also. And the optimum growth temperature is 37 degrees Celsius. In nutrient agar, we can see a large circular, low convex, grayish, white moist, and smooth opaque colonies. And in blood agar, we can see the colonies are very big and circular with grey and moist like appearance. In McConkey agar, the colonies are circular, moist, and smooth, and they appear flat and pink color. And in Muller-Hinton agar, we can see the pale straw colored colonies. In eosine methylene EMB agar, which is the specialized agar for E. coli, you can see the green metallic sheen colonies. That is speciality for E. coli in EMB agar. And in cysteine lactose electrolyte deficient agar, which is called as CLED agar, it gives lactose positive yellow colonies. In liquid media, the E. coli, they show homogeneous turbate, which is within 12 to 18 hours, that form the sedimentation on the bottom of the test tubes, that is called as pellicles. That sedimentation is called as pellicles. After prolonged incubation, that is greater than 72 hours, you can see the pellicles that are formed on the liquid media. These heavy deposits are formed which dispersed on shaking, so they may disperse. This is the hierarchy of this E. coli. It belongs to the kingdom of bacteria of phylum Proteobacteria, and class Gamma Proteobacteria, order Enterobacteriales, family Enterobacteriaceae, with genus E. coli, species E. coli. And coming to the pathogenicity of E. coli, there are 4 important strains of E. coli. The first one is enterohemorrhagic E. coli, which is called as EHEC, and enterotoxogen E. coli, which is called as ETEC, and enteroinvasive E. coli, which is EIEC, and enteropathogenic E. coli, that is EPEC. This enterohemorrhagic E. coli, it produces the cytotoxins which disturbs the protein synthesis within the host cells. These toxins are otherwise called as virocytotoxins or sugar-like toxins, and they are pathogenic to humans. And mostly this infection, that is enterohemorrhagic E. coli infection, that occurs via the fecal or oral route. The symptoms, it ranges from very mild diarrhea to severe bloody diarrhea. And the severe cases, it may lead to hemolytic uremic syndrome, that is called as a renal failure, and in advanced stage it leads to death also if it is untreated means. The common serotype of this enterohemorrhagic E. coli is E. coli O157H7. And coming to the enterotoxigenic E. coli, it is also known as traveler's diarrhea, as this E. coli is mostly present in the lower intestine of the warm-blooded animals. Most of the symptoms and diseases are related to the digestive system. So this enterotoxigenic E. coli is also known as traveler's diarrhea, and infection that leads to watery diarrhea which may last up to a week. It also includes the symptoms like abdominal cramps, and sometimes it may be nausea and headache, and it also produces diarrhea in children with dehydration also, and in sometimes cholera infantum, that is similar to choleral disease. And this enterotoxigenic E. coli, it also possesses colonization factors like antigens like pili antigen, K antigen, to enhance their virulence capacity. Enteroinvasive E. coli, which is transmitted through fecal or oral route, and the transmission occurs by following the ingestion of the contaminated foods, and the organism that invade epithelial cells of the intestine, resulting in the mild form of dysentery. So on set of ingestion, it results in the mild form of dysentery, and the illness is characterized by the presence of blood and mucus in stools of the infected individuals. The characteristic feature of this enteroinvasive E. coli is its ability to induce entry into the epithelial cells and disseminate from cell to cell. This infection can occur through contaminated food or water, or through mechanical factors such as flies. And enteropathogenic E. coli, it is also responsible for infantile diarrhea. It is also a diarrheal disease. The pathogenic infection has only recently developed, and it also adheres to the intestinal mucosa, and it causes loss of microvilli, and it prevents the entry of bacteria into mucosa. It also produces Shiga-like toxins. And diagnostics, lab diagnosis, we can test for identification, and like McConkie agar, it shows positive, indole also positive, methyl red reduction test also positive, citrate reduction test also negative, in lysine decarboxylate it shows positive, and acid gas production positive, and lactose, it also able to ferment lactose, and oxidase it shows negative. Treatments for this E. coli disease, mostly the healthy adults need to know treatment for E. coli infection, as their own immune system can prevent this symptoms as well. And in some studies, it has been noticed that the treatment with antibiotics increases the chances of getting HUS. So this effect occurs because antibiotic destroys the bacterial cell wall, which causes them to release even more toxins. When necessary, treatment includes the replacement of fluids as well as the electrolytes to treat or prevent dehydration. So that's all about E. coli. Thank you, students.